So we will start the third session. I'm just sharing the slides. Just move to that part. All right. So we will switch slightly now to a newer area of malaria biology. And I'll take you back to 1991. So, remember in my early presentation, in my first talk, probably when I showed you the parasite, I said that it has a single mitochondrion, it has a nucleus, it has a single mitochondrion, and it has an apicoplast. But it was only in the 1990s really that the apicoplast was discovered. And the story of the discovery of this organelle is also very interesting. So in 1991, Professor R.J. Meyer Nilsson's group and his collaborators were looking at the parasite genome and trying to, the genome had not been sequenced at the time. So the sequence of the parasite genome was not known. So they were trying to isolate the genome. And when they did pulse-field gel electrophoresis, they found that, there was this main nuclear DNA with 14 chromosomes, there was a nuclear genome. There was, in addition, another genome. And they initially thought that this was probably the mitochondrial gene. But when they purified the DNA and they actually sequenced it, they found that the sequence of this DNA, this was a circular genome that I show over here, a circular DNA molecule to the left that I just shown here, that circular closed double-stranded DNA. And when it was sequenced, it was understood that the sequence of this DNA sequence of this genome is not like mitochondrial genomes, but is very similar to plant plastid genomes or plant chloroplast genomes. And it has an inverted repeat, which is shown over here, which has a large subunit, small subunit RNA, and then the repeat in a different direction. With the small subunit, large subunit RNA, these are called IRA and IRB regions. Then it has RNA polymerase subunits, it has elongation factor TU, there's a SUF gene, which we now know to be SUF-B, and so on and so forth. And there are a lot of protein, there are a lot of ribosomal RNAs being encoded and also ribosomal proteins being encoded by the apicoplast, and also some tRNAs. So it has a very reduced genome. It's not as large as chloroplast genomes. It is only 35 kb. Chloroplast genomes, plastid genomes can be 135 or more. It's short, it's smaller, but in terms of its sequence and the genes present, it's very similar to plant plastids. Therefore, and if you look at the mitochondrial genome of the parasite of malaria, it's a very small genome. It's a 6 kb sequence and it's repeated. So it's actually stretches of 6 kb just after each other. Just encodes 3 proteins: cytochrome oxidase 1, cytochrome oxidase 3, and cytochrome B. Remember, I described cytochrome B as a target for that. And it has fragmented ribosomal RNAs being encoded. So very small genome, and it's distinct from the plastid. So essentially, when this genome was discovered, it was quite surprising because people had never thought that the malaria parasite, an intracellular human parasite, or an intracellular mammalian parasite, will have a plastid-like organelle. So the DNA of the organelle was discovered first, and then the organelle was identified in electron microscopy. So basically, the question was asked: have malaria parasites 3 genomes? And once the genome was identified as that of a plastid genome, the next question was: is the malaria parasite then a plant? Which is taking it a little far, it's not a plant, but basically, evolutionarily, you have to understand how the apicoplast is coming to existence. And this was a very interesting question posed by McFadden and Pollard in an essay in 1997. So we need to understand how this apicoplast has come evolutionarily and how the process of retention of a plastid has happened in the malaria parasite. So I'll go through this a little bit. So endosymbiosis, right? The process by which, if you take the mitochondrion, intracellular organelles might have evolved from prokaryotic forms. So if you look, so the theory of the apicoplast, of the evolution of the apicoplast and the fact that it is in the malaria parasites is also interesting because when you look at the apicoplast membrane, the apicoplast is surrounded not by 2 membranes, but by 4 membranes. You know that the mitochondrion is surrounded by 2 membranes. The plastids, green plastids, or the dairy plastids are surrounded by 2 membranes. But the apicoplast is surrounded, the organelle is surrounded by 4 membranes. So an explanation for this is secondary endosymbiosis. And I will briefly describe here what is meant by the process of secondary endosymbiosis. So early on, millions of years ago, there would have been an ancestral cyanobacteria, which is also called a cyanobacteria, which can make its own food, surrounded by these 2 membranes, which is engulfed by a phagocytic eukaryote, right? So this was engulfed and the cyanobacterium became part of this eukaryotic cell. Say a red algal cell, for instance, right? So here you have the membrane of the cyanobacterium, then you have the membrane of the red alga. If such a red alga ancestral cell was then engulfed by a second endosymbiotic event, this is the first endosymbiotic event, by a second endosymbiotic event of by a phagocytic eukaryote, then you have formation of such a cell, in which you will have the nucleus of the alga, you will have the cyanobacterial first primary endosymbiont, then you will have the membrane of the red alga, and then the phagocytic, then this surface membrane, the cell membrane of the engulfing phagocytic eukaryote. In such a way, now you will have four membranes surrounding the organelle. This particular nuclear genomes can get lost, they get converted into nucleomorphs and then completely lost. And much of the genes of this can also get migrated into the nucleus of the post over evolutionary time. Now, this is so basically you can have a secondary plastid-like structure, or secondary endosymbiosis event giving rise to a plastid-like organelle with four membranes. This is the process. So this missing link between what we currently see in non-green plastids, the process of photosynthesis has been lost. In the malaria parasite, the apicoplast does not have any genes associated with photosynthesis. So it doesn't need to photosynthesize. So the photosynthesis machinery has been completely lost. It's surrounded by four membranes, but it does some other stuff. It does not photosynthesize. There are intermediates such as in chromerids, chromerabilia, which have a partially endosymbiotic life on corals. And these organisms have retained photosynthesis. So they are also four membrane bound, but they have retained photosynthesis. And malaria apicoplasts organisms have lost photosynthesis. So this is what we are looking at currently in the malaria parasite. Complete loss of photosynthesis. An apicoplast does not have galactolipids, and these are non-green plastids. Right? So this is the organization of the apicoplast. So all the apicomplexan parasites, including not all, except Toxoplasma, Toxoplasma, Emeria, etc., they have apicoplast. So therefore, the apicoplast of the malaria parasite has become a delict plastid. It was a plastid, but it lost photosynthesis. It is a secondary endosymbiont, and it is a delict plastid. Why is it interesting also? So if you look at the entire evolutionary process, what we are saying is that a cyanobacterium was engulfed by a primary endosymbiotic event to give rise to a red alga, for instance, and the red alga got engulfed by another eukaryote, and this engulfment process is a secondary endosymbiotic event, and you had a secondary plastid in organisms like Chromera, which are chromerids and are rarely and survive on coral, live a independent semi-parasitic life on corals. They are photosynthetic. And similarly, other organisms that were became parasitized initially retained photosynthesis, but then this would have been, they retained photosynthesis here, but the process was lost as the parasites became obligate intracellular parasites. So apicomplexan organisms could be filating heterotrophs, then became photoautotrophs, and then became obligate intracellular parasites. Right? And organisms such as Plasmodium and Toxoplasma have the plastid, and Cryptosporidium is also an epicomplex and has lost the plastid. So there also loss of the epicoplast. So it's a very interesting phenomenon when you see you can have free-living heterotrophs, photoautotrophs, and obligate intracellular parasites, and the process has retained the organelle but has lost the capacity to photosynthesize. Now, what is the apicoplast doing, and why is the apicoplast essential? So it was shown that if you inhibit the replication of the apicoplast genome, initially by using gyrase inhibitors, if you use an inhibitor, say novobiosine or ciprofloxacin, which are antibiotics that target gyrases, then the apicoplast genome could not replicate, and since it could not replicate, the parasite was able to could not survive. So the apicoplast is essential for parasite survival. But why is it essential for parasite survival? And the interesting thing is that if it is essential for parasite survival, we know that mammalian hosts such as us do not carry the apicoplast or any plastid. So if the processes of this organelle are unique, can they give us targets for new drug discovery? Remember, my first part of the presentation was when we are looking for new drug targets. So is the apicoplast the Achilles heel by which new and can it function as a target site for new molecules? But what does it do? Why is it essential for the parasite if it does not photosynthesize, right? So it does other things. It does not photosynthesize. So Chromeravelia photosynthesizes. But if you come to the epicomplex and parasites, they do not photosynthesize. The epicoplast does not have photosynthesis. Toxoplasma and Plasmodium epicoplast carry out fatty acid biosynthesis by the FAST2 pathway. They carry out acetyl synthesis, which is really by the non-malonate pathway, which is essential. And it is this pathway that makes the epicoplast indispensable for parasite survival and growth. They can also synthesize heme. This is a bit surprising, but there is so much heme coming in from hemoglobin, yet the parasite has its own heme synthesis pathway. And this is because the parasite needs heme not only in the RBC, but also in the liver stages and the mosquito stages where you don't have RBC hemoglobin. So therefore, its own hemoglobin pathway, heme synthesis pathway is essential in those stages where excessive heme is not coming in from the RBCs. In Thylidia, there is isopenoid synthesis. In Cryptosporidium, there is no plastids, so none of these actually work. But in the malaria parasite, the fatty acid biosynthesis, isopenoid biosynthesis, and synthesis of heme become very major pathways which are important for the parasite. In addition, a paper that came out in 2017 showed a very interesting observation. It said that a key malaria metabolite modulates mosquito blood seeking, feeding, and susceptibility to infection. And what is this metabolite? In the isopenoid biosynthesis pathway, before the final product of the pathway, isopentenyl pyrophosphate is made, IPP is made, there is an intermediate called HMPP. And this intermediate, it was shown, can actually be sent out from the epicoplast through the parasite into the human RBC. And it causes an overexpression of carbon dioxide of aldehydes and monoterpenes, which work as chemoattractants for mosquitoes. So people who are infected and have, of course, the parasite which carries the epicoplast have this HMBPP production, which leads to higher levels of production of aldehydes, and therefore mosquitoes will be. So if you have a room full of 100 people, of which 1 or 2 people are infected with the malaria parasite, it's quite likely that mosquitoes will actually be attracted towards those individuals who are infected, who are malaria infected, because they will be expressing a higher amount of aldehydes and monoterpenes, which are chemoattractants for mosquito biting, and therefore the spread of infection will happen. So epicoplast therefore also seems to have a role in the transmission of malaria. Now to an infection in other individuals. So in terms of drug discovery and development, the epicoplast is also very, not only for its biology and for its other things which are of profound interest, but for people looking for antimalarials, it's also an interesting site to look at. And epicomplexans have retained the plastid because they are profoundly dependent on its metabolism, and the endosymbiont is therefore very important for parasite survival. And can this be used as can this become a liability for the parasite? Because can this now become a target? And can humans now discover drugs that actually target the parasite, because they can target the epicoplast, and the epicoplast is not there in the human. And because it's not there in the human, can we have specific molecules for specific epicoplast proteins which will not have safety issues for human use? Now, if you look at the parasite of the epicoplast morphology, that is also very interesting. It's not like it's a around a sort of discoid chloroplast. In the early stages, it's a very small organelle. But in the late trophozoid, early schizon stages, in the blood, this is a blood in the RBC, in the blood stages, it becomes elongated. It starts becoming branched. There's a single epicoplast and there's a single mitochondrion, and they are present in proximity to each other. And it becomes branched and elongated. And then as this parasite divides into merozoites, each of the merozoites not only get a nucleus, but also get a single epicoplast and a single mitochondrion. And here you can see that each of the merozoites is getting a single epicoplast. In the liver stages, and this is data from Walter Hughes's lab, it's very interesting. Here in green, you have seen the epicoplast. Here in red, you see the mitochondria. This is a single infected liver cell. Just look at the way the epicoplast is branched. Extremely highly branched, single organ. This is a single organ. Highly branched, like a web-like structure. Similarly, the mitochondria, single mitochondria, branched web-like structure. It's a sort of mesh. And then as division starts happening, and the merosomes and merozoites are formed, Then there is the apicoplast divides first. So the mitochondria has barely begun division. The apicoplasts have got divided first. And following apicoplast division, mitochondria get divided. And each of the merozoites get a single mitochondria and a single apicoplast. But look at the morphology of the single organelle, and how interesting the division is and how they can pan out in single organelles in these cells. Now, apicoplast, as you saw, has a very small genome, 35 kb genome, codes for a limited number of proteins, limited functions. So, how does this fatty acid biosynthesis, isopenoid biosynthesis, because of heme intermediates happen? There are a lot of proteins, many proteins. Around 1/6 of the, it's predicted that around 1/6 of the genome of the nucleus is actually apicoplast. It makes a protein that are targeted to the apicoplast. It might be fewer, but it's a very important component. So a lot of proteins that are targeted to the apicoplast, most of the proteins, there are exceptions, and I will not go into exceptions today, carry a signal sequence, which is where targets to the ER, and also carry a transit peptide. A transit peptide is not a fixed sequence, but it has certain features. So it's basically positively charged. And you can have algorithms that can recognize mutative transit peptides. So there's a signal sequence and a transit peptide at the N-terminus, and this is a bipartite targeting sequence that is required for transport of proteins that are encoded by the nuclear genome, transited in the cytosol, and then transported into the apicoplast for biological function. So basically, the genome itself is highly reduced, the apicoplast genome itself. It encodes important proteins, but most of the proteins that are important for function are nuclear encoded, and they are targeted to the apicoplast. Now, among the pathways in the apicoplast, I mentioned to you earlier 3 pathways. I mentioned to you the isopenoid biosynthesis pathway, the heme biosynthesis pathway, and the fatty acid biosynthesis pathway. In the genome of the apicoplast, circular genome, there is one gene for a protein called SUB, which is a protein that is important for assembly of iron-sulfur clusters on proteins. So this is a post-translational modification. Iron-sulfur clusters are assembled on protein molecules and have very important functional and catalytic roles. I will describe it shortly. Which told us that the sub-pathway iron-sulfur complexation is likely to be an important pathway taking place in the apicoplast. So let's move on to that. And through this, I will try and address 1 or 2 issues of drug targeting and development and the novelty of pathways. Now, if you look at iron-sulfur complexation, what is iron-sulfur complexation? Iron-sulfur complexes are complexes of iron and sulfur. They can be complexes that have 2 iron molecules and 2 Fe atoms and 2 sulfur atoms and make another complex. Or they can have 4 Fe, 4s, or they can have intermediates, 3 Fe, 2s, 4 Fe, 4 Fe, 3s, 2s, like. But the most common ones are 2 Fe, 2s, 4 Fe, 4s. So this is a structure which looks like this. This is a structure which is like a cuboid, and the cysteine and the iron atoms are present here. The sulfur and the iron atoms are present here. And these are coordinated further by cysteine residues on the protein. Okay. This is one of the iron-sulfur containing proteins. This is an example. And you can have cysteines or histidines that coordinate the iron-sulfur cluster on here. So this is the apoprotein. And if an iron-sulfur cluster comes and assembles here and coordinates with these 3 cysteines and this histidine here, then this will become a 4 Fe, 4s holo form. Right. Now, iron-sulfur clusters are not formed easily. There are assembly pathways. There are proteins that help in cluster assembly and formation of these iron-sulfur clusters. And there are 3 iron-sulfur cluster assembly pathways known. These are the ISC biogenesis pathway, the NIF pathway, and the SUB pathway. The ISC pathway for formation of iron-sulfur clusters is found in bacteria and eukaryotes, most of the eukaryotes. The NIF pathway is found in bacteria, in nitrogen-fixing bacteria, but the SUB pathway, that is what we are discussing now, is present in archaea, in bacteria, in protozoa, and in plants, but is absent in mammals. So humans, who are mammals, will not have the SUB pathway, but the SUB pathway seems to be present in the apicoplast, in the malaria parasite. Now, in the malaria parasite, my group had worked on this pathway, and we were able to show that the ISC and the sub pathways, so there are two parts. All of the proteins of these two pathways, ISC and sub, are encoded for by the nucleogene, except for sub B, which is encoded for by the apicoplast. The sub pathway partitions to the apicoplast, the ISC pathway partitions to the nucleus, to the mitochondrion. So you have each of these pathways in the mitochondria and the apicoplast in plasma. Now, the sub pathway is essential for apicoplast maintenance and parasite survival. Knockout studies have shown that if you knock out make a mutation in sub C, in it's an ATPase protein, it has ATPase activity, then the sub pathway, then the apicoplast maintenance is affected and parasites die. So the sub pathway, it was shown in 2013, is essential for parasite survival and apicoplast maintenance. Now, what is FES clusters? Why are they important? Proteins that carry FES clusters. So FES clusters are one of the most ancient post-translational modifications. They probably were into existence when the atmosphere of the Earth was reducing. As it became oxidizing, FES clusters became parts of internal proteins that protected from oxidation. They play a very important role in electron transfer reactions. They play an important role in redox and non-redox catalysis. So they're important for protein function. Some of them also play a role in the structural integrity, maintaining structural integrities of particular proteins. But to assemble these clusters, you have to have a whole pathway. What is the pathway? So if you take the ISC pathway or the sub pathway or the NIF pathway, there are some commonalities. The first commonality is that you require something for mobilization of sulfur from cysteine. Cysteine is a source of sulfur. You need an enzyme, which is a cysteine desulfurase, to mobilize sulfur from cysteine. And you also need a source of iron. Once the cysteine is mobilized once the sulfur is mobilized from cysteine, you have to assemble the iron and the sulfur on a scaffold. So you need a scaffold, protein or a scaffold complex, which assembles the iron sulfur cluster. And then you have to transfer this cluster onto the target apoprotein. So suppose you have 10 proteins that require iron sulfur cluster for their activity. Then the scaffold and the cluster must be transferred to these apoproteins either directly from the scaffold or through intermediary transfer proteins, right? It's an important step in the sulfur biogenesis and the FES biogenesis part. So we started looking at it. I'm just showing you some examples basically to tell you how the sub pathway operates and what were the functionalities of the sub pathway that could be interrogated and how can one interrogate this. So basically first identify genes of the sub pathway. So one has to look for homologs. So you look at bacterial sub pathways and you say, okay, E. coli has these sub S, sub A, sub B, sub D. Do we have homologs in the nucleus of the parasite? Yes, we do. Sub B is encoded by the apicoplast itself, but the rest of them we could find homologs in the nucleus. So these genes were taken out and they were expressed as recombinant proteins in bacteria, in E. coli, to purify. And then the reactions were assembled. Now, if you want to see whether the sub S protein, which was a putative cysteine desulfurase, can the subprotein convert cysteine into alanine? And by doing so, release a persulfide intermediate. And this release process is enhanced by interaction with sub C. So we use this assay to try and understand what was happening. And basically, if you see over here, if you have sub E alone, there is no desulfurase activity. Similarly, if you have sub S alone, there is nearly no activity. But as you keep increasing the concentration of sub E, right, the proportion of sub E to sub A, you start sub S, you find that there's an increase in the activity of PF, of the cysteine desulfurase. So cysteine desulfurase alone, sub S alone has low activity, but as you keep increasing the sub E, there is increase in activity till it plateaus. So sub E enhances the cysteine desulfurase activity of sub S. And by in vitro interaction, we could also show that these interactivities are each other. Now, this is a model, the structural model that tells you how this happens. So this is a paradoxal phosphate which interacts with sub S and helps in this whole process of the cysteine desulfurase activity. So this is paradoxal phosphate over here. This is the cysteine, this is the residue of PF sub S, okay, which interacts with PF sub E. And the transfer of the persulfide intermediate takes place from this to this, and sub E becomes a carrier for that transfer. And the interaction of sub E is very important for sub S to function as a cysteine desulfurase. the question we asked was, can we inhibit surface function? And by doing so, can we inhibit parasite growth? Because if surface is the cysteine desulfurase, which we were able to establish, and if surface interacts with it and enhances its activity, somehow, now there is no known inhibitor of surface. So even in E. coli, in bacterial systems, so far, there is no chemical inhibitor of surface known. So we decided to use a compound called D-cycloserine, which is an inhibitor that is used, that also inhibits Mycobacterium tuberculosis. And D-cycloserine was used because D-cycloserine is able to form an adduct with pyridoxal phosphate, with PLP. So what we were testing was, suppose we make a PLP adduct. So we were able to do that. So if you incubate PF surface alone, you will see that there is a PLP dependent peak. The moment you add DCS, D-cycloserine, the adduct is formed and the peak is shifted. So there was adduct formation. And when you do that in culture, when you give D-cycloserine in culture, it has a high IC50, yet but we could see that D-cycloserine was able to inhibit parasite growth. So something that forms an adduct with PLP can inhibit the activity of the enzyme and also inhibit parasite growth. So it was, in theory, one argued that it was possible to have surface targeted, perhaps not by using a PLP adduct, because PLP is also there in other enzymes. But in theory, you can target, you can form an adduct in this case, but you can also have perhaps got more specific small molecules that either inhibit surface activity or inhibit the interaction between surface and surface. And if we can do that, we can actually have new molecules against the surf pathway. People are working on these lines, but there is no real good inhibitor known so far. But because the surf pathway is completely absent in the human, it is essential for the malaria parasite. Knocking off SUF-C and later on surface, as I will show you now, show that it also knocks out the function of SUF pathway in the blood stages as well as in the mosquito stages. Makes it a very strong candidate for a target for antimalarials. Now, as I mentioned earlier, SUF-C had been knocked out by Giselberg's group and Giselberg et al. In 2013, and they had shown that if you knock out SUF-C, blood stage infection is affected drastically and there is parasite death. Now, so we tested our knockout for PF surface, and with my colleague here, what we did was we made knockouts, and these were SUF knockouts, state-specific knockouts. I'm not describing the processes, so basically it's a long process, but basically you have a knockout system which is state-specific. So you can have generate knockouts only in the sporozoid stages. So here you have the trap FLP system. So you can have, this is a surface knockout. This is a trap FLP background. Now, these knockouts were made, and when we made these knockouts and followed them in the mosquito, it was found that the wild type parasites, as expected, there were a lot of sporozoids in the salivary glands. But the surface knockout parasites had hardly any sporozoids in the salivary glands. And look at these images. In the normal parasites, trap FLP normal parasite background, you have these are oocysts. And as you can see in this oocyst and in this oocyst, so many elongated sporozoids eliminated. But look at the surface knockout parasites. There is no sporozoid formation. There is very retardation of sporozoids, small numbers, extremely small. And the number of salivary gland sporozoids is also reduced. So basically, it's seen that sporozoid development and maturation is deeply affected in the surface knockout parasites. And when you do an infection with these, whatever number of sporozoids you can isolate, you do an infection, you find that the pre-patent period, the time taken for appearance of the parasites from the liver to the blood is extremely long, right? So there was an elongation, and whatever parasites could survive were parasites that had lost, that were not knocked out. So basically, all the knockout parasites are not able to develop sporozoids, but there might be some that are not knocked out. So those limited small number of parasites can form sporozoids, and these can also cause infection, but the infection process is very slow because the numbers are very small. Okay, so all parasites in the affected mice are only those parasites that has escaped the excision process, the knockout process. So basically, we could establish the surface is essential, and if you knock out surface, then the growth in the mosquito stage is severely compromised, and you don't have sporozoid formation, and you don't see salivary gland sporozoids. So this also told us that if you inhibit the SUF pathway, not only are the blood stages affected, but the mosquito stages are also affected. So this is very good. This is good news, because that tells us that the SUF pathway, if you have an inhibitor ever, then you will also not only just inhibit the parasite, in the humans. But if the mosquitoes take in blood, and if with the blood they also take in the drug, because if a person has malaria and is being given an inhibitor or a drug of the sub pathway, then if the mosquito bite happens and with the blood meal the mosquito also takes in the drug, then the parasite, whatever parasite it's taking in, will not be able to survive in the mosquito. Right? So the whole question is that you can have it's a so it's important in multiple stages, and therefore inhibitors can be of use in multiple stages theoretically. Of course, one needs to do a lot of work to understand whether that will actually happen. Now the next stage, this was a cysteine desulfurinase, the first part. What is the scaffold, right? So by a series of experimentation, again, by making recombinant proteins, etc., and drawing identities from the bacterial surface system. I'm not describing the details here, but basically we could understand through a series of co-immunoprecipitation experiments where you can make an antibody against one of the interacting partners. And then from the parasite lysate, use that antibody to pull down not only the target protein, but also all the interacting proteins. And then see what the composition of the complex is like. Then you can also do cross-linking in vivo. So what you can do is you can put a cross-linker, a protein-protein cross-linker in the parasite, and then you can cross-link all the interacting partners and then purify the complex and then see what this complex had, right? So protein-protein cross-linking experiments would tell us the size of the complex, the components of the complex, etc. So by doing these kinds of experiments, we were able to show that you have this sort of complex in the parasite, the scaffold complex. You had sub B, you had sub D, and then B independently interacted with sub C and D independently interacted with sub C. So you get two molecules of sub C and one molecule each of sub B and D. Sub C is an ATPase, it has ATPase activity. And here what we could establish was that if you take sub C alone, it has ATPase activity, sub D alone has no activity. And if you do sub C and sub D together, the ATPase activity of sub C is greatly enhanced. So sub D interacts with sub C and enhances its ATPase activity. And this ATPase activity has roles to play in the assembly of the iron-sulfur complex on sub B. Okay, so this is the scaffold then, sub BC2D scaffold, and this assembles a 4-sulfur 4-iron cluster, and we could reconstitute this on this complex in vitro. When you do homology modeling to understand what sort of structures these might be, this is not a crystal structure or a cryo-EM structure or whatever. This is basically a molecular model modeled on the known structures of bacterial proteins. So this is the model for PF sub B, this is the model for sub D, and this is sub C. And look at this interesting thing here. We know that sub D is able to increase the ATPase activity of sub C, and it seems to do so by a loop-like structure here, which seems to integrate into a domain of PF sub C. So the logic we followed was, or tried to follow was, that if you can inhibit this interaction, then can you lower the ATPase activity of PF sub C, and can that affect the 4Fe4S cluster formation over here? So my colleagues of mine are now trying to see whether you can actually dock molecules and inhibit this sub D sub C interaction process so that you can have inhibition of scaffold assembly on this. So once the scaffold is assembled, you now need to transfer this cluster, which is already assembled over here, onto the target proteins. The question was, does this get transferred on its own directly, or do you need carrier proteins like these two mentioned here to transfer this cluster? And to cut a long story short, we were able to show that the scaffold does not directly transfer 4Fe4S onto a target protein like aconitrase, for instance, over here, but requires either NFU protein or SAFE protein to carry out the transfer, and these serve as carrier proteins to transfer the cluster. So finally, what do we know? So we know that the apicoplast has a subpathway, that only one gene of this pathway is apicoplast encoded. All the other genes have, during evolutionary time, been transferred from the genome of the endosymbiont to the nucleus, and now they are translated in the cytosol, and then they are targeted back into the apicoplast, where they perform important functions, which in this case is assembly of iron-sulfur clusters. And the surface component, together with SAF-E, it converts cysteine to alanine and generates a persulfide, which is the sulfur source for the iron-sulfur cluster assembly. We still don't know what the iron donor is, but iron nevertheless comes here. And sub-B complex then assembles iron-sulfur over here, and you have interaction of sub-C and sub-D. And sub-D can increase the ATPase activity of sub-C. And we can possibly have two intervention sites, one over here for sub-S or for the sub-S-sub-E interaction, and one over here for the sub-D or the sub-C sub-D interaction, because this is also essential and this is also essential for the sub-pathway to be functional. And as individual knockouts have shown, for both sub-S and sub-C, if you knock this out or knock this out, the parasite is not able to survive. This has been shown also in the mosquito stages, and this has been shown in the blood stages. The next is the transfer, and then you have transfer of the 4Fe4S cluster through sulfate or NIFU onto target proteins. One very interesting thing is here. What are the target proteins for the iron-sulfur cluster in the apicoplast of the malaria vaccine? You have ferredoxin and ferredoxin reductase. These are the target. You have DOXP pathway enzymes. Remember, I talked about the isoprenoid biosynthesis pathway. This is that pathway. ISPG and ISPA, the terminal enzymes of the pathway, require 4Fe4S clusters. If they don't have 4Fe4S clusters, they will not be able to convert, they will not be able to act. And therefore, 4Fe4S clusters are critical for their activity. And therefore, if you do not form 4Fe4S clusters and you inhibit the sub-pathway in any way, the isoprenoid biosynthesis pathway will not be able to function. So, and if it's not able to function, the parasite will not be able to survive. So this is a very critical, for the parasite. And therefore, I think the explanation of why this is happening and why inhibitors are working so well is because two enzymes of the isoprenoid biosynthesis pathway require FES clusters for that activity. You also have lipoic acid synthase, a very important enzyme that also requires FES clusters, and an adenosine tRNA methyltransferase MAIA-B, which also requires FES clusters. There might be more enzymes. In fact, we are discovering more proteins which are important for DNA repair that seem to carry 4Fe4S clusters. And these seem to be required not so much for activity, but for protein structure integrity and stabilizing protein structures. Also an interesting way that iron-sulfur clusters can work. So, but through this presentation that I made today, what I was trying to show you was that apart from the isoprenoid biosynthesis pathway, for which phosphonomycin is already being in trials as an apicoplast targeting molecule, there is possibility of other pathways such as sub-S, which are absent in the human, to be future targets for drug discovery and development. And people are working in this direction, and work from our lab and others has shown that this can be a good strategy, a very interesting strategy for targeting malaria. So I hope there were some issues raised in the session we had today, and that there would be many more questions in your mind and things that you could go and for. And if there are any questions that you would like to ask me, you can send me your queries on my Gmail account. It's saman.habib@gmail.com, and perhaps I could be able to help some of you if there are any very important questions that you would like to raise. Thank you.